Hello, this is Sky Marshall welcoming you to this Bite-Size Bio webinar, which today is sponsored by Mabtech. Today's presentation is titled Measuring Antigen-Specific Immune Responses at the Single-Cell Level and is being presented by James Perry, June Park, and Renata Varnaite. James began his career at Beth Israel Deaconess Medical Center and Harvard University, where he developed assays for the HIV Vaccine Trials Network's global initiatives. He then moved on to support discovery and translational work at Genosea Biosciences, focusing on personalized cancer vaccines. Currently at Elicio Therapeutics, James leads clinical immunogenicity studies for a lymph node-targeted KRAS cancer immunotherapy in pancreatic ductal adenocarcinoma. June serves as the R&D manager at MilliporeSigma. MilliporeSigma is the US and Canada life science business of Merck KGaA Darmstadt, Germany. He studied biomaterials and immunology at the University of Toronto and completed his postdoctoral work at Harvard Medical School, where he investigated small molecule regulators of beta integrins. Since joining MilliporeSigma in 2006, June has primarily focused on developing novel detection technologies and cell-based assays. Renata earned her PhD in medical science from Karolinska Institutet, Sweden in 2022. During her research, she used FluoroSpot assays to measure human T-cell and B-cell responses to infections and vaccines. In 2023, she joined Mabtech, where she assists customers in effectively applying ELISpot and FluoroSpot technology across various therapeutic areas, ranging from infectious disease vaccines to cell and gene therapies. As always, we will have a question and answer session after the presentation, so please type any questions that you have into the questions box, which appears at the top right of your screen, and I'll put them to our speakers at the end. Details of how to access the on-demand recording of this webinar will be sent to you by email shortly. So now over to our speakers for the presentation.
Thank you for the introduction. I'm very excited to be speaking today on behalf of Mabtech to discuss how ELISpot and FluoroSpot assays are used in clinical trials. Today we'll focus a bit more on FluoroSpot use, but both of the assays are excellent tools for detecting antigen-specific T-cell responses at a single-cell level. So if we look over the last 25 years into clinical trials that use T-cell ELISpot or FluoroSpot assay, you will see that those assays have been very popular and their use have actually increased over the years, especially during the COVID-19 pandemic, as illustrated here. There are more assays to monitor T-cell immunity, and some other popular assays include intracellular cytokine staining and AIM assays. However, you can see that ELISpot have been even more popular than those flow cytometry-based methods. And there are typically a few reasons why ELISpot is so popular. But the three main reasons are that these assays are sensitive, they're versatile, and they're very easy to perform. And this is what really made them the gold standard for detecting antigen-specific immune responses. You can detect T-cell and B-cell responses using this assay, but in this picture here, I'm showing a single-color ELISpot and a triple-color FluoroSpot assay. So ELISpot assay is using enzymatic reaction to amplify the signal, and in FluoroSpot, we are detecting the fluorescence-based detection systems. Both of these assays can be used in many different contexts, and they typically have a very high agreement between each other. However, of course, they're using different technologies and different assay principle, so they typically are not used within the same study, and they're not always giving identical results, but the trends and the responses that you're seeing in patients should be usually very consistent between the two methods. So you might ask, why would I use ELISpot or FluoroSpot assay instead of other sandwich assays like ELISA or a bead-based multiplex assay? There are multiple reasons for that, but the primary reason is that when you're using ELISA or multiplex, you're looking at the secretion after it has already happened. You're also looking at bulk level of secretion, and you can't identify how many cells in your sample are secreting that cytokine of interest. So if in your assay you want to know the number of cells that are secreting the cytokine or the antibody of interest, that's where ELISpot and FluoroSpot assays excel. And with that single cell resolution, you also get increased sensitivity because even low levels of secretion from a single cell can be detected with these methods. So let's go through the step-by-step of how you would set up a typical T-cell ELISpot assay. In this case, you would start with 96-well plates that you would pre-coat with a capture antibody targeting the cytokine of interest. So you can see here that we are incubating your immune cells, which are often PBMC samples or splenocytes from mouse. And you would incubate those cells with a specific stimulus. That could be usually peptide pools or whole protein. During that incubation, if there are any T cells that are responding to the stimulus, they will start secreting the cytokines. And if those cells are secreting the interferon gamma cytokine, that secretion will be captured by the capture antibodies. You would then remove the cells and use the detection antibody that's conjugated to an enzyme to detect the footprint of secretion. The last step would be to add a precipitating substrate. And this is a time-sensitive step, so the longer you incubate the substrate on the plates, the more signal you will amplify. But the resulting readout will look something like this. Each of these individual spots represent a single cell that has secreted interferon gamma. And the readout is typically expressed as numbers of spots per well or numbers of spots per million PBMCs or so. FluoroSpot assay, in contrast, is using fluorescence for detection of the spots. Otherwise, FluoroSpot is very similar to perform as ELISpot assay. It's just as easy to perform. It's not taking a longer time to actually get it done in the lab. So it's not a more complicated method, even though a lot of people sometimes think that fluorescence is more tricky somehow. So in the biggest benefit of using FluoroSpot is that you can actually multiplex cytokine detection. So in this case, I'm showing an example where we're detecting interferon gamma, IL-2, and TNF alpha. And we're using 3 different detection systems to distinguish the secretion of the 3 cytokines. If you look at the image here, you will see that we have many different spots, and there are 3 different colors corresponding to interferon gamma, IL-2, and TNF secretion. So that's already giving you more data from a single well compared to interferon gamma ELISpot assay. But in addition to getting the total numbers of spots, you also can look at co-localization of signal from all these 3 cytokines and be able to detect and enumerate polyfunctional T cells. Polyfunctional T cells are the cells that secrete more than one cytokine at once, and they've been widely studied in different disease contexts, and they have been investigated as a correlate of protection. And I think it's primarily been studied in the HIV field, but also a lot during the SARS-CoV-2 pandemic and a lot in cancer immunotherapy more recently. Another area where polyfunctional readout is relevant and being used quite a lot is tuberculosis. So there is a diagnostic test available for tuberculosis, and it's called T-Spot, and that's interferon gamma ELISpot assay. There are, however, multiple publications that has come out recently showing that if you are, instead of looking at just interferon gamma, if you also look at polyfunctional T-cell responses by interferon gamma, IL-2, and TNF FluoroSpot, you may have an improved sensitivity and accuracy in diagnosing TB, as well as in some cases distinguishing active TB from latent TB. So this is really a good example of how polyfunctional T-cell detection can be very useful in diagnosing infectious diseases. If we now think of a broader view of how these assays are used in clinical trials across different clinical trial bases, if you were to go into clinicaltrials.gov right now and search for ELISpot and FluoroSpot, you will find around 200 active clinical trials that explicitly mention ELISpot and FluoroSpot use within primary, secondary, or other endpoints and throughout different phases. And what we can see from this table here is that the primary use of ELISpot and FluoroSpot is within the secondary outcome. It's sometimes also used in the primary outcome and exploratory outcomes, but the secondary really dominates. And the primary therapeutic areas where these assays are used is cancer immunotherapy, infectious disease, autoimmunity, and transplantation. So I have showed in the first slide today that ELISpot has been used very widely. So you might ask, what is the added benefit of FluoroSpot? So I want to show you a few examples of how FluoroSpot have historically been used in clinical trials and what is the added benefit of FluoroSpot. If we start with infectious disease vaccines, you often would like to measure wanted and unwanted immunogenicity after you administer your vaccine to patients. The typical combination would be to look at Th1 responses, which are typically antiviral responses, something that you really want to induce in an infectious disease vaccine. But then you might also want to look at Th2 responses, something that you want to avoid. They often can lead to allergic reactions. So to do that, you could combine detection of both Th1 and Th2 cytokines with FluoroSpot assay. I want to highlight this paper, which is describing a phase 2 trial of SARS-CoV-2 vaccine. In this specific trial, they have used dual color FluoroSpot assay of interferon gamma and IL-5 to measure T-cell responses after immunization. And what they found is that the vaccine has induced a very robust Th1 response, as measured by interferon gamma, but they didn't detect any IL-5 response, which is really great news for a vaccine trial, because that can give you some confidence that hopefully there will be minimal or no allergic reactions in the patients that receive the vaccine. If you're, for example, studying fungal diseases or especially autoimmune conditions, you may also want to include cytokines like IL-17. And I want to show you an example of a monoclonal drug therapy in type 1 diabetes, where they've used a interferon gamma and IL-17A and IL-17F FluoroSpot assay. In this case, they have administered the monoclonal drug to type 1 diabetes patients, and then they followed T-cell responses after administration of therapy. And what they found was that the treatment selectively decreased these IL-17 secreting T-cell frequencies, but there was no change in interferon gamma responses over treatment. So that really showed that if you were to only look at interferon gamma, you may have not seen an effect that your drug has on the patient population. And the last example I want to show you is regarding cytotoxicity assessment. We know that a lot of viral infections like HIV, but also, especially in immuno-oncology, we want to induce that cytotoxicity with our therapies. And I want to show you this paper that looked at personalized mRNA cancer vaccines. And they tested the patients first with a interferon gamma ELISpot assay to identify responders to the neoantigens. And once they've identified those responders, they wanted to confirm that the responses induced by the neoantigen vaccine were indeed polyfunctional. So they used interferon gamma and IL-2 and granzyme B FluoroSpot to check that there was indeed polyfunctional T-cell response, and they have indeed confirmed it. So this is also a great way of showing how strong and robust is T-cell response in your patients after cancer immunotherapy. What we're seeing in biopharma industry, we at Mabtech are definitely seeing an increased uptake of FluoroSpot in clinical trials, but there is also an additional trend that we are seeing, and that's automation of this assay. As you know, in clinical trials, you're dealing with a high number of samples, and you want to make sure that your data is reproducible and consistent, especially if you're doing multi-site testing. So automation can really help with that. And I want to highlight a paper that came out very recently from a vaccine research center at NIH, where the researchers have automated the entire FluoroSpot assay, from the cell seeding all the way to data acquisition on the Mabtech iris reader. And you can see that they have, with this process, they have achieved really excellent precision for this assay. And with cell-based assays, you're typically it's typically harder to achieve high precision, but these precision values are indeed excellent. And as you can see, the researchers themselves are saying that this would be an excellent platform for clinical trial endpoint testing, but also for any other high throughput application where you need automation. So with that, I would like to pass the microphone to June, who will talk about one of the most critical reagents that you require to be successful with this assay, and that's the 9612 FluoroSpot plates. So thank you for listening.
Thank you so much, Renata. That was a really good overview. Hello, everyone. My name is June, and together with Yasmin, who is the product manager for our multi-screen plates, again, we would like to welcome everyone. Quick overview of the topics that I'll be covering. I'll start with the different membrane types that can be used for ELISpot applications. Then I will cover the different plate designs and formats. Last, I will go over our Immobilon FL membrane for fluorescent applications and other considerations for ELISpot assays. Membrane really is the critical component of the ELISpot assay. Please do not try this, but if you ever tried ELISpot assay with regular ELISA plates, you probably notice that there are no spots, right? This is because high protein binding and retention, particularly that of capture antibodies, are critical for robust spot formation. And this in turn requires microporous membranes with vastly increased surface area. However, not all microporous membrane substrate have desired affinity for protein binding. This bar graph shows various microporous membranes in regard to protein binding. Here we use radio-labeled IgG to measure protein binding. Binding is indicated on y-axis. Different membranes are indicated on x-axis. Due to high surface area, given a right surface chemistry, some of these microporous membranes exhibit very high level of protein binding. Overall, nitrocellulose and immobilon PBDF membranes show highest binding. And this high level of protein binding is critical for sufficient amount of capture antibodies to bind to the membrane in order to achieve good ELISpot result. And that is why both membrane types are preferred membranes for ELISpot assays. However, the interplay between protein binding and retention does create difference between nitrocellulose and immobilon. Let's take a look. This slide shows direct comparison between nitrocellulose and immobilon. The microporous structure of the membranes can be seen with scanning electron microscope images. In both membrane types, you can readily see greatly increased surface area for protein binding. However, next to the scanning electron microscope image, we also see actual ELISpot counts. Here we're looking at interferon gamma response, and we see that the spot counts are higher on immobilon compared to nitrocellulose for the reasons that we described earlier. As mentioned in the previous slide, this is due to the better retention of the bound antibody on PBDF membrane, leading to overall increase in spot counts, essentially making it more sensitive. This makes immobilon PBDF useful for conventional ELISpot assays, as well as a B-cell ELISpot assay, where antigenic proteins need to be captured on membrane surfaces. Now, saying that, while we prefer immobilon PBDF, if you're happy with nitrocellulose membrane, we recommend that you continue using nitrocellulose. Now let's consider plate design and formats. We offer ELISpot plates in two formats, the Classic and HTS. The Classic plates were introduced originally for ELISpot applications, but based on the customer feedback, we launched HTS plates specifically to address high throughput applications and automation. Each format is available with both membrane types, and the key differences are the design of the underdrain and sidewalls. Looking specifically at the automation-compatible HTS slide plates, these are certain features that I would like to highlight. It has rigid sidewalls to enhance handling, and this also reduced edge effect compared to classical plates. New underdrain configuration reduces evaporation and leakage during assay. The underdrain also helps to keep the membrane flat for more even cell distribution and attachment and subsequent spot formations. The plates are offered in white and clear plate frame, compatible with all imaging systems, and we can also put barcodes upon request. While I cover the HTS plates, I want to quickly go over this slide since we had a number of customers asking if the underdrain need to be removed. If you have used these plates recently, you probably noticed that the removal of underdrain can be difficult. Historically, we recommend removing the underdrain before drying the membrane for imaging. However, recently we have reduced the gap space between underdrain and membrane in order to eliminate liquid trapping between the two. This makes it difficult to manually remove the underdrain, but since liquid trapping is eliminated, there is really no need to remove the underdrain before drying the membrane. We recommend drying the plates overnight with the underdrain and then imaging them. Similar to membrane options, from a customer perspective, question we often get is which plate type is preferable? And again, similar to the membrane options, if you're currently using a particular plate type and happy with it, please continue using it. However, if you're starting something new, here are a few considerations. The graph shows side-by-side performance comparison between membrane types and plate types. Bar graph on the left side shows the results from nitrocellulose membrane. The bar graphs on the right side shows results from immobilon PBDF membrane. Within each membrane type, we also show results from classic versus HTS plates. In summary, immobilon PBDF clearly offers higher spot counts. This again goes back to higher, tighter protein binding, particularly the capture antibodies mentioned earlier. In addition, the HTS format offers tighter CV values due to the reduction of edge effect and flatter membrane. Taken together, HTS plate means more spots with less variability. Another form factor that I want to showcase is our 8-well strips with reusable membrane frame. Similar to 8-well ELISA strip plates, these plates offer convenience of using 8 wells at a time, so you can use only what you need and save the rest of the plate for later uses. Okay, let's move on to the last section of the presentation, fluorescent application and other considerations. As Renata mentioned previously, fluorescent ELISpot brings multiplex capability to ELISpot assay, which in most traditional cases been done as a singleplex. But in order for ELISpot fluorescent application to work, it is critical that the membrane has low autofluorescence. This concept should be quite familiar with customers that do fluorescent Western blots. And in fact, the membrane that we recommend for fluorescent ELISpot is a membrane that we recommend for fluorescent Western blotting applications. Shown are fluorescent imaging of different membrane types from left to right. We're imaging the membrane at typically used green, yellow, and red channels from top to bottom. Colors are pseudo-colors, but clearly show how immobilon PBDF membrane exhibit high level of autofluorescence, which results from light scattering. Nitrocellulose does show reduced light scattering, but still exhibit noticeable level of autofluorescence, particularly when green channel is used for fluorescent imaging. Now, however, if you look at immobilon FL membrane, it shows no or negligible level of light scattering. In fact, the open people think that we did not put a membrane to scan here because you really don't see anything. But membrane is there. And the reason that you don't see any background fluorescence is because this membrane has extremely low light scattering property. What this translates to ELISpot assay is that immobilized FL membrane offer best signal-to-noise ratio when it comes to spot counting. On the right side, you can see how regular immobilon compares to immobilon FL when wells are imaged using fluorescent ELISpot reader. Again, it looks as if there is nothing there, since the membrane has negligible light scattering. Essentially, this reduction in background fluorescence is what enables detection of fluorescent spots with high sensitivity. Currently, immobilon FL membrane plates are available via Mabtech, but we also offer custom plates with FL membranes upon request. The last few slides, I would like to cover some tips and tricks that we've been hearing from our customers. First, if you encounter aggregates due to cell deaths when using frozen PBMCs, we highly recommend using benzonates to break down the nucleic acid strands which are causing the aggregates. Typically 50 units per milliliter with 30-minute incubation at room temperature works really well. Second, if you experience non-specific spot formation on your control wells with blank media, it is worthwhile to filter your bioattenuated secondary antibodies. The bioattenuated secondary antibodies can denature over time, leading to protein aggregation and phobic interactions with the membrane. Simply filtering the 0.2 micron syringe filter can help in this case to remove any antibody aggregates. Please remember that we have a lot more information on our website, and I really encourage everyone to check it out. Simply type in ELISpot and it'll lead you to related information. Thank you so much for your time today.
Thank you to Chun for sharing your insights and for passing things off. Today, I will present From Theory to Practice, lessons from a cancer immunotherapy program, where I'll share our optimization of the interferon gamma granzyme B FluoroSpot and discuss its implementation for immunomonitoring in our phase 1 Amplified 201 trial. This slide contains an important cautionary note regarding forward-looking statements, as well as statements as to endorsements. Here's today's agenda. I'll start things off by reviewing the mechanism of action of our ELI-002-2P monotherapy and discuss the design of the phase 1 Amplified 201 trial. Then we'll move on to selecting and optimizing the interferon gamma granzyme B FluoroSpot, and I'll wrap things up by sharing the Amplified 201 immunogenicity findings and their correlation with the clinical outcomes we observed in the trial. Let's start with our therapy's mechanism of action and the design of the Amplified 201 study. Pancreatic ductal adenocarcinoma and colorectal cancer are two of the most lethal malignancies worldwide due to several factors, including late-stage diagnosis, rapid progression, and challenging tumor microenvironments. There are limited therapeutic options available following the standard local regional course of treatment, and relapse is unfortunately common due to micrometastatic disease, resulting in roughly 3 to 5 year survival for metastatic disease. G12D and G12R mutant KRAS driver mutations are commonly expressed across both indications and throughout disease progression. These mutations are also necessary for tumor survival, which makes them an ideal target for immunotherapy and an attractive option for off-the-shelf treatment. ELI-002-2P is just that. It is an off-the-shelf clinical stage lymph node targeted mutant KRAS specific amphiphile immunotherapy for adjuvant treatment of pancreatic and colorectal cancers. It's composed of 3 components, consisting of 2 amphiphile modified long peptides for G12R and G12D mutations, thus the 2P designation, which are both combined with an amphiphile modified TLR9 agonist CpG 7909. Traditional cancer immunotherapy has shown variable success and poor clinical efficacy, largely due to its inability to effectively stimulate patient immune systems. The small molecular weight of conventional peptides and adjuvants results in their distribution within the systemic circulation after immunization and ineffective delivery to secondary lymph node organs where immune responses are orchestrated. ELI-002-2P overcomes these barriers by utilizing an albumin-binding lipid to hitchhike the therapeutic components to the lymph nodes via binding to endogenous albumin. Accumulation of the therapeutic components within the lymph nodes results in their preferential internalization by resident APC. These resident APC then coordinate robust immune responses by stimulating, in this case, mutant KRAS-specific T cells. Preclinical studies of this approach demonstrated enhanced efficacy through potent reprogramming of the immune microenvironment, increasing T cell expansion, tumor infiltration, and the induction of memory T cells, underscoring the potential promise of the lymph node targeted therapeutic approach. The Amplify 201 trial was a phase 1 adjuvant dose ranging study to assess the safety, efficacy, and immunogenicity of our monotherapy. It was given after tumor resection to patients who had no radiographic evidence of disease, but had minimal residual disease by circulating tumor DNA or serum tumor antigen biomarkers. We hypothesized that the administration of ELI-002 after tumor resection and within the setting of minimal residual disease would induce expansion of mutant KRAS-specific T cells, ultimately increasing the potential for anti-tumor activity, controlling micrometastatic disease progression, and delaying radiographic recurrence. There were 25 subjects enrolled with confirmed G12D or G12R tumor mutations across 5 dose cohorts. Each cohort received a fixed dose of AMP peptides 2P and one of the 5 escalating doses of AMP CpG 7909, which range from 0.1 to 10 mg. Patients received 6 doses over an 8-week period as part of the prime series of immunization, followed by a 4-dose booster 3 months after the prime. The study endpoint that we will focus on today will be the mutant KRAS-specific T cell responses elicited by ELI-002, along with the attributes of the interferon gamma granzyme B FluoroSpot that supported the longitudinal assessments made within the study. Now that we've covered the mechanism of action and trial design, I would like to share how we selected and developed the FluoroSpot assay for use in this trial. So there were several program requirements we needed to address to ensure a robust investigation of the immune responses generated by our monotherapy. Luckily, many of the technical characteristics of the FluoroSpot align with these requirements, making it an ideal choice for the sensitive longitudinal assessments needed. First, we needed a high throughput method that could rapidly measure antigen-specific T cells across multiple antigens in several patient samples and be amenable to automation. We chose to use human PBMC because they provide a readily accessible means to comprehensively evaluate our therapy's mechanism of action and how it modulates patients' antigen-specific T cells throughout the course of treatment. Our PBMC for the trial were limited, so we wanted to ensure that we got the most out of our analysis by finding an assay with low sample requirements, like the FluoroSpot, but one that could also support robust methods such as technical replicates, ensuring the accuracy of our measurements. There was a requirement for a quantitative assessment of ELI-002 immune responses, and the data output needed to be directly comparable to other data sets, either over time or to other values from other studies. The FluoroSpot measures the magnitude of response by reporting spot-forming cells, so it fit within this criteria and was well aligned to the requirement for comparable longitudinal assessments. We preferred a conventional assay for this program, which offers several benefits, including an extensive literature base on optimization and standardization, as well as the ability for regulatory bodies and the broader scientific community to readily understand our methods. And the last box we wanted to check was finding an assay that could accommodate compliance with our good clinical laboratory practices and be validated. Having resources on this topic within the literature and provided by vendors was a plus for us. So the FluoroSpot checked all of the technical boxes for our immunogenicity program, and its scientific characteristics also gave us confidence that they could be leveraged to demonstrate the potential of our immunotherapy. FluoroSpot is a highly sensitive and functional assay because it captures multiple secreted analytes at the single cell level over time. Therefore, it can identify rare T cells that otherwise would be difficult to detect with other methods, which sometimes require elaborate expansion protocols or manipulation of your cell populations. The assessment of polyfunctional T cells was also important, as they have been shown to strongly correlate with better clinical outcomes in traditional vaccine studies and adoptive cell therapies. And the FluoroSpot's ability to analyze up to 4 analytes in a single well, along with a wide range of available assessments, gave us the flexibility to select a protocol tailored to our specific goal of investigating cytotoxic effector T cells elicited by our therapy. As mentioned previously, the conventional nature of performing this assay also supported its selection, allowing us to easily investigate immune responses not only to immunizing antigens, but also to other common mutant KRAS antigens, and in some cases, patient-specific neoantigens. The FluoroSpot is available in a wide range of species-specific formats, which we utilize for our development that I'll get into greater detail about on the following slides. We began our assay optimization using a mouse interferon gamma ELIspot, as study-specific PBMC were not immediately available, and they would be best used for our clinical evaluations. We utilized a preclinical immunization protocol to generate mutant KRAS-specific immune responses in mice against our therapeutic antigens. This assay was used to identify key assay parameters with the greatest impact on assay optimization to ensure robust and highly reproducible data. We started these characterization studies by examining media types that would support cell health and function without increasing our assay background, as having a low assay background is critical to maximizing signal-to-noise ratio and the sensitivity of your assay by ensuring that rare antigen-specific T cell responses are not masked. We performed multiple experiments using 3 available serum-free media and evaluated the mean interferon gamma spot-forming cells in DMSO negative control wells. In the top figure, we observed a similar background with 2 of the media evaluated in these experiments, and 1 of the media showed highly variable increased responses under our experimental conditions. Cell density and peptide pool concentration were also evaluated alongside these media investigations, as both variables directly affect the ELIspot's ability to detect responses. Mean interferon gamma spot-forming cells were evaluated utilizing a combination of 3 different splenocyte concentrations and 3 different peptide pool concentrations. We found that too many cells, shown in green on the bottom figure, hinder the accurate detection of quantification of antigen-specific cells, most likely due to cell stacking and high background noise observed under these conditions. A high concentration of peptide, in this case 5 micrograms per mil, also blunted observed responses across all 3 cell densities and produced the lowest magnitudes of interferon gamma spot-forming cells in this experiment. We continued our assay characterization studies by examining additional variables, including the size of overlapping peptides within each of our peptide pools, the length of peptide stimulation, and the effect that varying the number of assay replicates had on the variability of the data we produce. All of this would be important for controlling inter-assay variability before moving on to studies evaluating inter-operator variability and final implementation in human studies. Once we had a strong foundation with the mouse interferon gamma ELIspot, we used these findings to identify variables critical for optimization in the human assay, given their high contribution to assay variability and reduced sensitivity. Our final studies in mice, which gave us confidence to move into human experiments, assess mutant KRAS-specific responses to immunizing antigens G12D and G12R, as well as responses induced by ELI-002 to 5 other mutant KRAS antigens commonly expressed in pancreatic and colorectal cancers. Those are G12V, C, A, S, and G13D. Because this assay will be performed by multiple operators throughout the study, we evaluated the robustness of our methods by assessing inter-operator variability. We wanted to gain insights into the potential for differences in measurements generated by multiple operators when evaluating cells from the same mice. We found that responses were comparable when different operators perform assay setup, development, and data analysis, and this gave us confidence to perform a final mouse experiment to increase the magnitude of responses captured by our assay through peptide pool optimization. We investigated whether changes in amino acid length in our peptide pools could improve the magnitude of responses observed and sought a pool optimal for detecting both CD4 and CD8 positive antigen-specific T cells. 2 peptide pools were evaluated that contained varying amino acid lengths for the 7 mutant KRAS antigens shown in the bottom figure. And we observed superior magnitude responses for all 7 mutant KRAS antigens with our optimized peptide pool when compared to the original peptide pool when using the same splenocytes. These findings underscore the importance of identifying and optimizing key assay reagents before using human samples that are often limited. The characterized mouse assay was moved into optimization studies with human samples, where we continued to refine the assay background, improve response magnitude, and finalize our methods for the clinical trial with limited use of our human PBMC. The primary parameters that contributed to the assay variability in our human studies were sample quality and media type, as well as cell density, peptide pool concentration, and the size of the peptides within our pools as observed in the mouse. One of the benefits of the FluoroSpot assay is its multiplexing capabilities. We wanted to utilize this advantage to have a more comprehensive profile of the immune responses elicited by our immunotherapy. The FluoroSpot facilitated these assessments without increasing PBMC use and enabled us to observe polyfunctional T cells throughout the course of treatment. Interferon gamma and granzyme B were selected for our human trial to investigate cytokine-mediated effector and cytolytic functions, as they are key mechanisms of cell-mediated anti-tumor activity. First, we wanted to ensure that translating our findings from the mouse and human ELISpots would not compromise our ability to detect robust responses when implementing the beneficial multiplexing technology of the FluoroSpot. In the figure to the right, you can see the results of a comparative study performed using the mouse interferon gamma ELISpot and FluoroSpot assays, in which we observed a strong positive correlation in the detection of interferon gamma producing mutant KRAS specific T cells. The mean response for multiple mice to each one of the 7 mutant KRAS antigens is shown in the figure, with an R-squared value of 0.9924, giving us confidence in the comparable detection of interferon gamma spot-forming cells by the FluoroSpot. A strong positive correlation was also observed for human studies on interferon gamma, as well as those evaluating granzyme B, highlighting the robustness of the FluoroSpot when properly characterized and optimized for your needs. An additional benefit of the FluoroSpot assay was observed during our protocol development, where decreased subjectivity for operators resulted from the elimination of the visual-based enzymatic development required for ELISpot. This helped us ultimately standardize and further refine our methods for the clinical study. Next, I wanted to highlight that throughout these optimization studies, we also evaluated the implementation of automation and specialized equipment to ensure consistent results. We observed that high-quality, pre-coded plates yielded reliable readouts during longitudinal assessments of our patient samples, thereby streamlining our protocol by eliminating the need for a pre-coding step. We also developed a protocol using an automated plate washer and stacker to support high-throughput efforts and hands-free plate washing. The HTS engineered plates were easy to use with our stacker and plate washer, and the washer protocol optimization for these plates was very straightforward to implement. We used the Mabtech Iris for plate reading because it is Part 11 compliant and intuitive, which aligns well with our GCLP work for immunogenicity assessments. The raw spot algorithm utilized by the technology allowed us to easily adjust settings for spot size and intensity during optimization of our data analysis, as well as change the brightness and contrast for added operator confidence during these method standardization studies. The range of settings within the technology gave us confidence in our finalized standardized protocol, ensuring identification of rare T cells and robust, repeatable evaluation of our patient samples. Once our human assay was optimized, it was really wonderful to see the fruits of our labor support project goals for the Amplify 201 trial. And now I'll share some of the data for a single subject from this trial. The optimized human ex vivo FluoroSpot lets us evaluate the specificity, magnitude, and polyfunctionality of patient samples throughout treatment with high confidence. And below is the immunogenicity data generated for patient number 11 from the study. In the figure on the left, we observe a 2 to 12-fold increase after prime immunization to the individual peptide, mutant KRAS peptides listed within the figure. And the figure on the right highlights the changes in the functional profile of these mutant KRAS-specific T cells, not only showing an increase in the frequency of interferon gamma and granzyme B secreting cells, but also a notable increase in the subset of polyfunctional T cells secreting both of these analytes. The flexibility of this assay allows us to make multiple assessments for a single mutant KRAS antigen over time and generate a comprehensive immune profile for our ELI-002 amplified T cell responses by patient. The FluoroSpot's ease of use, high throughput capability, and low sample requirements enabled a robust, sensitive, longitudinal assessment of immune responses, which we were able to draw correlations to the trial's clinical outcomes, which I'll highlight on the following slides. Now I'd like to share the immunogenicity findings from our Amplify 201 trial and their correlation to the clinical outcomes we observed. In the Amplify 201 study, we assessed PBMC from 23 evaluable patients for specificity, polyfunctionality, and antigen breadth using direct ex vivo FluoroSpot and ICS assays. Patient samples were evaluated for mutant KRAS-specific responses to the 7 mutant KRAS antigens listed within this figure. And within this slide, you can also find the immunization schedule for all 5 patient cohorts in this dose-finding study shown in purple. Below that, you can see the longitudinal collection of PBMC used for our immunogenicity evaluations, where PBMC were first taken at baseline for each evaluable subject, and then 2 samples were taken in the middle of the prime series at weeks 1 and 4. Additional collections were performed 2 and 10 weeks after prime, prior to the administration of 4 booster doses at weeks 20 through 23, and as in the prime series, additional samples were collected within the booster series and 2 weeks after. Response definitions for the ex vivo FluoroSpot and ICS assays are shown to the right. We set criteria that a 2-fold or greater increase over baseline, along with additional assay-specific criteria indicated a therapeutic response. For the FluoroSpot, the post-treatment time point must show a greater than or equal to 50 spot-forming cells per million PBMC, along with the 2-fold increase mentioned previously to be considered a response. So here's the first set of summary data for our Amplify 201 trial. And it was really wonderful to see such high fold increases of mutant KRAS-specific T cells to our monotherapy, which is overall great news for the patients within the study. We observed expansion of mutant KRAS-specific T cells in all 5 cohorts by both ex vivo immunogenicity assays, and 84% of patients showed a mutant KRAS-specific T cell response following ELI-002 2P treatment. On the right, you can see a figure for the maximum response fold changes, where there was an average fold increase in mutant KRAS-specific T cells of 58.6 over baseline. I want to note that memory T cell expansion was observed alongside de novo priming of mutant KRAS-specific T cells after ELI-002 administration. This overall enhances immune surveillance of micrometastatic disease for this cohort of patients with minimal residual disease. On this slide, I would like to highlight the magnitude of our spot-forming cells identified in the ex vivo interferon gamma granzyme B FluoroSpot. The optimized assay captured max values that range from 61 to over 4,300 spot-forming cells for responder patients in a patient population with diverse HLA. 95% of immune responders responded to the immunizing antigens G12R and G12D, while 52% of those patients responded to all 7 mutant KRAS antigens evaluated within the study, demonstrating a wide breadth of immune response after immunization. To wrap things up today, the final analysis of our trial showed a strong correlation between relapse-free survival and the magnitude of T-cell response captured within our ex vivo assays. Evaluable patients were stratified into 2 groups for the assessment of relapse-free survival based on T-cell fold change max response, and patients who had a max response of 9.17-fold or greater over baseline had an 88% reduction in the risk of progression or death. Median RFS for the above threshold group had not been reached by data cutoff, while the median RFS for the below threshold group was 3 months. So today we've explored the importance of method development, optimization, and standardization of our ELI 002-2P trial and how those efforts set us up for success in this study. These experiments are ultimately important because they can help identify key assay parameters that make your optimized assay more robust and ensure alignment with program goals. And I've also discussed how our implementation of automation, as well as specialized consumables such as the pre-coded FluoroSpot plates and HDS plates, supported our protocol development and highlighted the benefits they've provided to standardization of our assay. Amplify 201 used the multiplexing technology of the FluoroSpot to comprehensively evaluate therapeutic immunogenicity, giving us a well-rounded view of cytokine-mediated effector and cytolytic functions of our patient mutant KRAS-specific cells, and its low sample requirements allowed us to capture twice as much data without increasing sample requirements. And finally, the interferon gamma granzyme B FluoroSpot ended up being a very powerful tool for investigation of immune correlates that we found within our clinical endpoints within the study, and it overall supported the mechanism of action of our therapy in a robust and repeatable format. With that, I'd like to thank the patients and their families for their participation in our trial. I'm grateful to my colleagues for all of their hard work in conducting these studies, and I want to thank Mabtech and MilliporeSigma for the opportunity to collaborate and present the findings from this phase 1 trial. And with that, I will happily take any questions.
Okay, thank you very much, James, June, and Renata. That was a great set of presentations. So we have a few questions from the audience. If anyone else has a question, please feel free to post it in the questions box that's at the top right of your screen. So I'll dive in. I've got a question for James. Is there some guide that I can follow to validate a FluoroSpot assay for clinical trial use?
Yeah, so thank you for the question. And yeah, there are actually many guides or publications out there. Many highlight, validation of bioanalytical assays, and many of those are provided by the FDA or other regulatory bodies. And they're ultimately going to give you a general consensus on how you can ensure that your assay is fit for purpose and can produce results that are accurate and consistent. The resources that I personally have found most helpful that don't come from regulatory bodies are authored by a scientist named Sylvia Janetsky. She has decades of experience with ELISpot and FluoroSpot, and a number of her publications, they range from very beginner to more high-level considerations for things such as assay validation. Her guides are honestly a wealth of information that help to inform our assay development program from its initial development to the optimized form that I was able to present today. A second resource that I can think of too is also actually found on the Mabtech website. They've done a very good job of consolidating and highlighting some of the key considerations for validation of ELISpot and FluoroSpot assays. And in that guide, you should be able to find links to some of the references that I've mentioned, those authored by Sylvia, as well as those that are provided by the FDA and other regulatory bodies.
All right, fantastic. Thanks very much. Another question, this one's for Renata. What are the differences between FluoroSpot and intracellular cytokine staining assays with flow cytometry? Do they yield similar data output?
Thanks, Guy. This is a great question. Both assays measure cytokine secretion or production, so they're quite similar in nature. You're looking at T cell activation via a readout of cytokine production or secretion, as I say. But there are quite a lot of differences. Intracellular cytokine staining is usually require a lot more expertise to operate the flow cytometer, and it also only looks at a snapshot in time during the stimulation. While with FluoroSpot, from our customers, what we've seen is that they can detect secretion or detect T cell responses throughout the entire stimulation period. So even if you have different cytokines that you're measuring and they have different kinetics when it comes to responses, they can detect that with FluoroSpot without any issues, and they're not limited to that time period. It can go as long as 48 hours or longer if required sometimes for different cytokines. So yeah, and then of course, depending on the sample input that you have, so if you're working with very precious samples or maybe pediatric samples or cancer immunotherapy patient samples, then FluoroSpot has this advantage of using fewer cells and also looking for rare T cell populations. You can use FluoroSpot for that purpose as well. So they're quite similar assays, but there are pros and cons for each.
Okay, thank you very much, Renata. I've got a question for June as well. So I think I missed it earlier. Why do you see more spot counts on PVDF membrane compared to nitrocellulose membrane?
Oh, yeah, sure. Yeah, that's a really interesting question. I think the key difference really boils down to the phobic interaction. So when any proteins interact with the substrate, they undergo denaturation. So usually the phobic cores of the protein gets exposed, right? And what happens is that in case of captured antibody, same thing happens. The phobic core gets exposed, and then it creates that phobic interaction with PVDF membrane, and that's a really strong interaction. I think the best example of that is when we're cooking eggs, right? When we cook eggs, egg proteins gets denatured, and that creates that phobic interaction. And we all know how difficult it is to get the eggs off the frying pan, right? So that phobic interaction retains the capture antibody during subsequent washes, and that indirect basically enables to see more spots on PVDF membrane.
Okay, excellent. Thank you very much. So we'll move on to another question. So this could be for June, but actually, in fact, I think maybe James could take this one. So it's please suggest how to reduce background, for example, with PBMC in media without antigen.
Okay, yeah, thank you for your question as well. So in an experimental condition of PBMC in media, without antigen, there could be a number of things that could lead to high background. The things that I would initially look at would be optimization of first the number of cells that you have in each well, right? You want to ensure that you do have cell-cell contact, but you don't want so many cells in the well that they're unhappy. That could ultimately lead to some of the background that you are seeing. You know, also considerations for the preparation of your samples. We found this immensely important in the development of our program also. You know, things like implementing an overnight rest of previously cryopreserved cells can ensure that, you know, the cells that are, you know, headed towards death after cryopreservation, they have time to ultimately die off before you can then wash them clean and then place them into your assays for assessments. I'm unsure if the individual who asked the question is using these wells as a negative control. And so a consideration also with that would be if you are, so in our case, we are using peptides reconstituted in DMSO. If you are using a high concentration of DMSO, that could ultimately make your cells unhappy. and then thus lead to higher background. I know that high levels of DMSO also interact with the membrane of the FluoroSpot plates as well. So that's another consideration. And then the last thing I would say is probably to make sure that you are filtering your reagents during the development of your FluoroSpot. You can just make sure that you don't run into any aggregates that could ultimately negatively affect the data.
Okay, fantastic. Thanks very much, James.
I could also add one more point on this,
Oh, yes.
on this question, because it comes up quite a lot. So I think James have mentioned a lot of the key considerations to reduce background. But also, depending on which cytokine you're working with, some of them just have inherently higher spontaneous levels of secretion. And that's typically the culprits could be granzyme B or TNF alpha. We do see sometimes that they have just higher levels in the unstimulated controls compared to, let's say, interferon gamma. And then one more consideration would be the sampling time point of the patient, and if the patient is undergoing any active infection or inflammation, then in those cases you may also be able to see some elevated spot numbers in the negative controls. So I just wanted to add these 2 points too, that sometimes it can be a confusing result if you get that.
All right, fantastic. Yeah, thanks for jumping in there. Of course, these questions, any of you can answer them if you want to. There's just time for one more question, and this person has said, fantastic presentations. So for ELISpot and FluoroSpot plates, what are the dangers or potential problems with using vacuum manifold to quickly drain and dry as a last step before reading for manual processing in low throughput studies?
Yeah, I can chime in.
Yeah, sorry,
Sorry. I was on mute. Yeah. So I think it's very difficult because what vacuum will do is that it'll actually affect the flatness, the membrane flatness of the membrane. So if you apply vacuum, membrane is not necessarily flat, right? And once you don't have a flat membrane, that in turn is going to affect ability to, count spots, right? Image them accurately. So that is why, in general, people do not use vacuum manifold for ELISpot assays. It's air dry, typically overnight. You preserve that flatness of the membrane, and that is actually critical for imaging and subsequent counting of the spots.
All right, fantastic. Well, that's a very interesting question to end on. So that brings us to the end of the webinar. Thank you again, James, June, and Renata, for your informative presentations and the great discussion here at the end. Thanks also to our sponsor, Mabtech. And finally, thank you to the audience for taking the time to attend and to ask questions. So until next time, good luck in your research, and goodbye from all of us at Mabtech and Bite Size Bio.